am Rinsha Rishad, studying in 9th standard in Kutubuzaman English Medium High School, Chammal. Today I am going to take 8th standard biology class for you. Our first chapter name is Life Mysteries in Little Chambers. This chapter is about cells, so let's go through it. Firstly, we can discuss about cell theory. Cell theory was formulated by M.J. Frieden and Theodore Strand, summarizing the findings of various scientists. It put forward two main concepts. Number one, the body of all organisms is made up of cell. And number two, cells are the structural and functional unit of all organisms. As we all know that we can't see cells using our naked eyes. We can observe it using microscope. So next, let's observe the parts of microscope and its uses.
Microscope's parts and their uses. A. The eyepiece, ocular, is the part of the microscope closest to your eyes, through which you look. It contains the ocular lens, which makes the image produced by the objective's lenses larger. An eyepiece may be labelled with its magnification or power, for example, 5x or 10x, meaning it enlarges 5 times or 10 times. B. The body tube holds the eyepiece in position over the revolving nosepiece and the objectives. The body tube may be straight up and down or slanted. C. The arm is the curved metal piece that holds the body tube in place over the stage and the base. D. The revolving nosepiece holds the objectives and allows you to change objectives while looking at a slide or sample. E. The objectives are the parts at the bottom of the body tube closest to the sample you are examining. Each objective has a lens and a tube-like holder, the mount. The longer mounts hold the stronger lenses, the high-power lenses. The shorter mounts hold the weaker lenses, the low-power lenses. Like the eyepieces, objectives also come in various powers. Microscope have several objectives, for example, 4x or 5x, 40x or 45x, and 100x. Some microscopes only have 1 or 2 objectives. F. The stage is the flat surface on which you put your slides or samples. G. The stage clips holds the slide in plate on the stage. H. Beneath the stage in all the besto microscope have a condenser having standard diameter of 39.5 mm, which collects and concentrates the light before it passes through the sample. I. The iris diaphragm is used to control the diameter of the cone angle of the light transmitted onto the specimen. J. The coarse adjustment knob is the large knob used to adjust the position of the body tube, allowing you to quickly bring your sample slide into view. K. The fine adjustment knob is the small knob used to change the position of the body tube by making small adjustments to the focus of your sample. Almost all of our besto microscopes have both coarse and fine adjustment knobs, but some have only one knob. L. The mirror, located beneath the stage diaphragm, increases the amount of light shining through your sample or slide. Sometimes we use lamp plugged into an electrical outlet instead of mirror or reflector. M. The base of the microscope is the heavy bottom part. It supports all the other parts of the microscope. N. Substage adjustment knob is used to move condenser up and down.
Now we observe the microscopic parts and their uses. Now let me ask you a question, please. Who was the first scientist who observed the cells? It was Robert Hooke, the first scientist who observed the cells using a simple microscope. He observed a section of cork through a microscope and called the tiny chambers as cells. Do you know what is meant by cell biology? Cell biology, study of cells, is an extensive branch of science which is rapidly developing nowadays. It is called cell biology. Now let me tell about the functions of cells. Number 1, resist foreign particles. Number 2, release energy from nutrients. And number 3, synthesis biomolecules. Once again we can say resist foreign particles, release energy from nutrients and synthesis biomolecules. Next we can observe different parts of a plant cell. Now we can say the plant cell organelles. Number 1, endoplasmic reticulum. Number 2, ribosome. Number 3, mitochondrion. Number 4, vacuole. And number 5, Golgi complex. Once again we can say endoplasmic reticulum, mitochondrion, ribosome, vacuole and Golgi complex. Now observe the parts of nucleus and their functions. In cells of organisms, where nucleus is not seen, such organisms is called prokaryotes. The organisms which they have a well-defined nucleus covered a membrane is called eukaryotes. Here we understood that we are included in eukaryotes. Next we can discuss about plastids. Plastids are the cell organelles that are exclusively found in plant cell. There are 3 kinds: chloroplast, chromoplast and leucoplast. As we all know that photosynthesis is performed by chloroplast. Chromoplast impart colors to fruits and flowers. Leucoplasts are the plastids having no specific color. The leucoplast is more seen in food materials. Now we can observe animal cell. So our chapter is over and thank you.